Hi everybody, welcome to our lab today on the inheritance of marshmallow critters. So we are going to explore how genes are inherited from parent to offspring, looking at traits that exist in marshmallow critters. So without further ado, I'm going to introduce you to our mom and dad. And for this, you're going to kind of jot down what the traits are. You have the handout that goes with this, and so this is somewhat of an interactive lab, even though we are not together as a class. So not for nothing, this is easily the cutest lab that we do. So let me introduce you to our marshmallow critters. So here is Mama. And as you can see, Mama has pink ears, she has blue eyes, and she has a nose. She has one back bump that's there. She has clear legs and a black curly tail. So she is just stinking cute if you take a peek. So there's Mom. Dad looks like this. So Dad is a little bit similar in that some of his traits he also has very similar to Mom's, in that he has pink ears, blue eyes and a nose. He has a single back bump and a black curly tail, along with clear legs. However, you can see that Dad here has some slight differences as well, because that's just kind of how we all work, right? We all have some differences, yet we all have some similarities in the traits that we exhibit. So for our marshmallow creatures, you can see how Mama here, her ears stand straight up, but Dad's kind of curled down. Dad has these wicked cool stripes. Mama is a curly cued, like she's got polka dotted swirlies that go on with her. So, that's her right there. But yet they do share some traits. So we are going to spend some time looking today at only 7 of the traits found in our little marshmallow creatures here. And those are the ear color, okay? We've got that as a trait. You also have their eye color as a trait as well. And you have whether or not they have a nose, the shape of their tail, the color of their tail, as well as the color of their legs and how many back bumps they have. Because out in the marshmallow creature world, you can have different forms of these genes, different forms of these alleles, okay? If you look at your chart, you'll see that ears come in the colors of white, red, and pink out in the critter gene pool. There are either 2, 1, or no back bumps as far as phenotypic traits that exist. Tails can be black or white. They can be curly or straight, and the legs can have either clear or they can be multicolored. So those are some of the traits that exist. Eyes as well, they can have either blue or they can have white eyes. And their nose, either they have a nose or they don't. That's kind of, as close as it gets. They either have one or they don't. So what we're going to look at is we are going to take Mom and Dad here and we're going to cross them so that we get a massive litter of little marshmallow critters. And to do that, though, we have to take their genes and their chromosomes, and their chromosomes have to go through splitting up in meiosis to create the gamete that each parent's going to give. And then the bringing together of those gametes and fertilization will tell us what the combinations are for our baby critter. So that's what you are going to do. You are going to actually determine what the genes are based on random chance, that your critter gets. So under normal circumstances, you would get this envelope here, and inside this envelope has all these strips, and you're going to see these strips kind of put up on the screen here. But each of these strips are, not only do they have different lengths, and we've got 7 sets, because this is the only thing we're going to focus on, okay, are these 7 chromosomes that these particular traits exist on. So you've got strips that are of the same length, and then you've got pairs that are different lengths. And so we have 7 of those. Not only that, on each one, you will see that there are letters. okay? And there is a capital letter and there is a lowercase letter, and these designate the alleles that the parents have. So you're gonna wanna write this down. Mom and dad, for the 7 traits only that we're looking at that I mentioned earlier, okay, mom and dad are both heterozygous for all of those traits. There is also the sex pair of chromosomes that we have here as well. Okay, so we don't want to forget those. We've got the sex pair of chromosomes, and the sex pair, right, determines whether or not it's a girl or a boy. So we have XX, okay, so in Marshmallow Critter World, XX is a girl, so that's mom's. And then in the red one, I'll find for the sex pair, if I pull those suckers out, I'm gonna find an X and a Y, okay? So that X and Y designate dad here, okay? So when we go through this process, it's not only are gonna be what are the traits that your baby critter get, but is it a girl or a boy baby critter? So we're gonna look at that too. All right, so here's where things get fun. And what you're gonna need is, you're gonna have your sheet where you have all of the traits laid out, okay? And what you guys also need is a coin, something you can flip, okay? Because you don't have these right in front of you to grab and, you know, randomly choose. So we're gonna have to kind of do this a little interestingly. All right, so here's what you're gonna do. Just to start, okay, we know that I'm gonna start with mom, okay? So I have mama here, okay? And mama has to make her egg that's going to fertilize with dad's sperm to make your baby critter. So in order to do that, remember, every one in meiosis, every one of her pairs separates, okay? So she is only going to donate one of each of these pairs, right? So here's another pair, right? It'll split, they separate. So I only get one of each length strip that mom will donate. Dad, same thing. All of his will split, leaving me with 1/2 of every pair. Okay, and then what we're gonna do is we're gonna bring those together. So for example, if I look at this first one, if I take mom's first pair, which is heterozygous, okay, for the A. So when you look on your sheet, you'll see what that codes for, right? Now, normally, this is a completely random process known as independent assortment, okay? And the way that a pair splits doesn't dictate the way another pair splits. So these pairs are normally face down, okay? And you kind of mix them up so you don't know which one, okay? So I pull them apart to represent meiosis. I put one away. Cool. I would do this for all 7 sets that I have of the, you know, chromosomes. You'd also do it for the sex pair too, so I guess technically 8. I find in dad's the same thing, right? There's dad's chromosome pair, okay, with those genes on them. Same thing, right? We mix them up. We pull them apart. Put one away. Once I have these for all, in order to create your critter and find out what baby has, we fertilize them, and these two come together. There's your fertilization. So you've got mom's contribution and dad's contribution. So when I flip those over and I look, my baby critter is going to be homozygous for ear color, okay? Homozygous, capital A, which if you look on your sheet would mean that my baby would have red ears. That's interesting, isn't it? So red ears. So I am going to do that for all of them to represent what would my baby look like when we bring all 7 or 8 pairs of those together to look at all 7 traits plus whether or not baby is a girl or a boy. So here's where you guys are going to have a little bit of say, so we can say, hey, this is a totally randomized process. With your coin, what I want you guys to do is to take these, right? And so we will see that I've got one chromosome, again, going back to the A one, that has the capital and the lowercase. All 7 of the traits that we're looking at, mom and dad are heterozygous, so they all have one of each. What we're gonna do is call A heads on your coin, call the capital letter heads on your coin, the lowercase letter is going to be tails. Okay, so we're going to do this, and then what you are going to do is grabbing your coin, okay, penny, dime, nickel, quarter, doesn't matter, okay, grabbing your coin, I want you to, for the first set, which is the A, and I just want you to follow right down, you're going to flip your coin. Okay, mine landed on heads. So I'm going to take care of mom's first. So mom is going to keep, she's going to give to baby the capital A. Okay, I'm going to do the same thing again for the next one. Okay, and whatever it is on your sheet, I'm just grabbing right now the ends, because that's the next biggest set of chromosomes. Okay, and I'm going to flip it again. It landed on tails. So I'm going to keep the recessive one. So I'm going to write that down. Mom's going to give that. So I'm going to go through all of these. Okay, when you get to the sex pair, okay, same thing, flip a coin. Only thing is, you're going to notice mom has 2 X's. No matter what, she's giving an X to her kid. So I'm going to keep an X from mom. I'm going to put the other X down. So what I'm doing is determining the set, okay, of alleles that mom would give to her kid. Once I do that, I'm going to do the same thing for dad. Okay, take your coin, flip it. Heads, dad is giving the dominant allele. Okay, put the other one away. Okay, so that's all you're going to be doing, is you're going to write that down. Okay, so hopefully this is making some sense as you do it. But your coin is like you, if you were here in school right now, you would be just randomly putting them upside down and randomly, mixing them up to pick just one out of the 2 to give to baby. So once you go through all 8 chromosome sets, flipping your coin, okay, you're going to write that down. What is mom contributing? What is dad contributing? Okay, for each one of those pairs. And then we're going to match them up. So one of the things you're going to want to make sure of too is with dad has 2 different chromosomes on his sex pair. So we're going to use heads for X, we're going to use tails for Y. Okay, so go ahead and flip that coin. Okay, whichever one you just got, that's the chromosome dad will give, okay, to baby. So put that one down on your sheet, okay, there. So when I flip my coins, okay, I'm doing that randomly. Again, you're doing the same thing. You might get something different than what I flipped because, hey, everybody got a 50-50 chance of what, right, it lands on. And since mom could give a capital A, could give a lowercase a, it's 50-50 which one she gives to baby. Once you have them all written down, okay, here's what dad's contributing in his gamete, what mom's contributing in her gamete, match your letters together. Okay, so for example, my A's, right? Okay, baby's going to have big A, little a. Okay, so now I look at that and say, okay, what is the phenotype that big A, little a codes for, and what will baby have for ear color? And I look at the chart that tells me what each genotype codes for, and it turns out that if I'm homozygous, okay, for the capital A, my ear color is red. So baby's ears for me are going to be red. You could have flipped it and gotten 2 little A's. If that's the case, your baby's ears are white, and you're going to write that down. So you go through all of your pairs. Okay, flipping that coin, figuring out what mom can give, flip the coin, figure out what dad can give, and you're going to match them up. Once you have that done, what we would like you guys to do is using mom and dad as a model. Okay, so this is kind of what they look like. Okay, and I'll put a picture up as well. Okay, now they are going to have maybe some other traits that we're not exploring, right? I mean, just like humans, we have 25-ish, plus or minus, okay, 1,000 coding genes that code for the traits that we have. Only for this exploration are we going to explore 7 of them, okay, plus whether they're a girl or a boy. And so in that, once you've written those down, we want you to try to draw out, what do you think your baby will look like? Okay, using mom and dad as a model, as well as the, the list of all of the traits that they have. Okay, and we want you to draw one out and share it with your teacher. Okay, share that picture on your document, because we're going to look at our class set, okay, of our class litter of baby critters, and we're going to look at the similarities and differences that will exist, even though they're all going to be siblings and they're all coming from these same two parents. Okay, and to show how important this process is to promote genetic variation in the next generation. So that's the goal within this lab. How does genetic variation happen when you have two parents, okay, to donate their chromosomes to the next generation, okay, which is really pretty cool, because otherwise we'd all be the same, right? So this is a wicked important piece to the idea of why nature has gone the way, if you will, of utilizing contributions from two parents into the next generation, so that we're not all identical. Now, of course, if you have an identical twin, okay, you are a genetically identical to that twin. But that was because when mom and dad's egg and sperm fused, okay, it was one zygote. And during those meiotic divisions, okay, that ball of cells pulled apart just a little bit. And when they pulled apart, okay, they got too far apart. And in doing that, it created two individuals who are genetically identical to each other, but as we know, two very different individuals, which is a pretty cool thing, with all the, identical twins I know in my life and have taught. Okay, you guys are all pretty cool. So, that's just something to remember. So we are going to instead look at all the possibilities of siblings that can come, even when we only look at 7, 8 different pairs of genes, and all the different possibilities that can exist from that. So this is just a random, event. Again, let the coin do the talking for you. Flip that coin, okay, for each pair, okay? And just to kind of review and reiterate, for all of the pairs, heads will be the dominant allele, tails will be the recessive, with the exception of the sex pair, okay, for dad, because mom can only give an X, so that's all you got to choose from. For dad, heads will be the X chromosome, tails will be the Y. And then we'll take a look at all of them. I'm pretty excited to see all of the different possibilities that you guys come up with, and hopefully we'll see through this lab how this works and give you guys an opportunity to get a little bit hands-on during this time. I have to say, not for nothing, I miss you guys. All of my students, I know all of the other bio teachers miss you guys. It's an interesting time we're in. We thank you all for your flexibility and the work you guys are doing through this process. So remember though, if you have any questions, please, please, please do not hesitate to contact your teacher, reach out, and work through the things that you need to get done, okay, so that you have the best opportunity you can in figuring out how this all works. So with that said, I'm gonna take mom and dad and we're gonna say our goodbyes, okay? We'll have the pictures here. Remember, come back to this anytime if you have any questions or concerns about how it works. But good luck, and we'll see you guys later.